This screencast discusses organelles. This topic may be found in Chapter 3 of your textbook. This screencast was designed to help you achieve the following objectives. Describe the structure and functions of each organelle. Describe the structure and functions of each of the three components of the cytoskeleton. In the previous screencast, you were given a general description for organelles. Organelles literally means little organs. They're specialized structures that perform specific functions within the cell. They're dispersed within the cytosol of the cytoplasm. Organelles are to the cell what your organs are to your body. Typically, cells have multiple copies of most organelles. Some organelles are surrounded by membranes, while other organelles have no membrane. First, let's talk about the organelles known as ribosomes. Ribosomes are composed of protein and ribosomal RNA. Their components are synthesized at the nucleolus and then moved through the nuclear pores into the cytoplasm. There, they are assembled into ribosomes, and either the ribosomes remain free in the cytoplasm or they are attached to the rough endoplasmic reticulum. Ribosomes are the sites of protein synthesis. The process known as translation occurs at the ribosomes. Ribosomes that are free in the cytoplasm produce proteins that remain in the cell. Ribosomes attached to the rough endoplasmic reticulum produce proteins that are destined to be secreted outside of the cell. Before discussing another organelle, I'd like to briefly discuss vesicles. Vesicles are spherical structures whose walls are composed of the same material that the cell membrane is composed of. They travel through the cytosol on tracks composed of proteins that form structures called microtubules. Vesicles are formed from membranous organelles or the cell membrane, and ultimately these vesicles are returned to membranous organelles or the cell membrane. A very important organelle of the cell is the endoplasmic reticulum. An endoplasmic reticulum literally means network within the cytoplasm. The endoplasmic reticulum consists of fluid-filled tubules called cisternae, which allow substances to be modified and moved throughout the cell. There are two types of endoplasmic reticulum. There's the rough endoplasmic reticulum that is studded with ribosomes, and the smooth endoplasmic reticulum that has no ribosomes and whose cisternae are more tubular in shape compared to the rough ER, where the cisternae look more like flattened tubules. The rough endoplasmic reticulum is studded with ribosomes. Protein manufactured by the ribosomes enters the cisternae of the rough endoplasmic reticulum. There, the proteins are modified, placed in transport vesicles, and these vesicles then migrate either to the plasma membrane or to the Golgi apparatus. This figure shows the process by which proteins are manufactured and put into transport vesicles. At position 1, amino acids are being bonded together and a protein is being formed. That protein enters the cisternae of the rough endoplasmic reticulum, number 2. There, the protein is modified and then is placed in a transport vesicle, number 4. And then that transport vesicle transports that protein either to the plasma membrane or to the Golgi apparatus. The role of smooth endoplasmic reticulum has nothing to do with protein synthesis, transport of protein, or modification of protein. Lipids, such as fatty acids and steroids, are synthesized in the smooth endoplasmic reticulum, and it's also the site for detoxification of harmful substances such as drugs, pesticides, and other potentially toxic substances. Since the liver is involved in the detoxification of substances, liver cells tend to have large numbers of smooth endoplasmic reticula. Smooth endoplasmic reticula are also involved in the detoxification of ethanol, the edible alcohol. Individuals who drink regularly tend to have large numbers of smooth endoplasmic reticula in their liver cells, and that gives them a resistance to intoxication. The Golgi apparatus is an organelle that is found in close proximity to the smooth and rough endoplasmic reticulum. Like the smooth and rough endoplasmic reticulum, it is composed of fluid-filled tubes called cisternae. The Golgi apparatus receives vesicles from the rough endoplasmic reticulum. Those vesicles contain protein, and once those vesicles are incorporated into the walls of the cisternae of the Golgi apparatus, the proteins that they contain are modified and packaged and eventually placed back into vesicles. Those vesicles leave the Golgi apparatus and are either secreted outside of the cell as the vesicle merges with the plasma membrane, are incorporated into the plasma membrane as the vesicle merges with the plasma membrane, or the vesicles are sent to locations within the cytoplasm. So you can sort of think of the Golgi apparatus as the UPS of the cell. This figure shows the various fates of vesicles packaged by the Golgi apparatus. In pathway 1, we're looking at a secretory vesicle where proteins packaged in this vesicle are going to be secreted outside of the cell as the vesicle merges with the plasma membrane. In pathway 2, the components of the wall of the vesicle are incorporated into the plasma membrane when the vesicle merges with the plasma membrane. In pathway number 3, the protein being packaged into this vesicle are digestive enzymes, and the organelle, which is formed by the packaging of these digestive enzymes in a vesicle, is called a lysosome. So lysosomes are vesicles. Vesicles formed by the Golgi apparatus that contain digestive enzymes. And the function of these lysosomes are to digest things. Some of the things that they digest are worn out, non-usable materials within the cell. For example, a worn out mitochondrion would be digested by a lysosome, and its components would then be used to assemble new mitochondria. Some cells, white blood cells, are able to engulf bacteria and other harmful microbes or toxins, and the lysosomes then dump their digestive enzymes onto these engulfed objects and digest them. Peroxisomes are organelles that destroy harmful substances. For example, they detoxify alcohol and they neutralize free radicals. Free radicals are molecules that have an unpaired electron. And the problem with free radicals is that they will steal electrons from other molecules, destroying those molecules in the process. And if those molecules are a part of a cell, they literally can destroy the cell. So free radicals present a definite danger to the health of the cells of the body. And free radicals are produced by cells as a normal byproduct of metabolism. Peroxisomes help neutralize these free radicals and also destroy other harmful substances. And in the process, they produce hydrogen peroxide, H2O2. And that's where peroxisomes get their name. This equation illustrates the detoxification of a free radical by peroxisomes. Peroxisomes use oxygen to detoxify free radicals, which are symbolized by the RH2, to produce a neutralized free radical R and hydrogen peroxide H2O2. Now, H2O2, hydrogen peroxide itself, is potentially toxic. That's why we use solutions of hydrogen peroxide as antiseptics. The body can use that hydrogen peroxide to destroy other toxic substances, or it can break it down into water and oxygen. If you've ever used hydrogen peroxide as an antiseptic, you of course know that when you pour it on an injury, it bubbles up. That's because there's an enzyme called catalase that breaks that hydrogen peroxide down into water and oxygen, and as that oxygen escapes, it causes effervescence or bubbling up. Proteasomes are barrel-shaped organelles that are composed of protein. Their function is to seek out, capture, and destroy proteins that are no longer required by the cell or that have been synthesized incorrectly. Mitochondria are these sausage-shaped organelles that are responsible for the production of ATP. Mitochondria use oxygen to break bonds of certain molecules, which releases energy. That energy, some of it is lost as heat, some of it is used to produce ATP. As you know, ATP is the energy currency of cells. It is what the cell uses almost exclusively as an energy source. For that reason, mitochondria are often referred to as the powerhouse of the cell. So the mitochondria uses oxygen to make ATP, and a byproduct of ATP production is carbon dioxide. So aerobic respiration occurs in the mitochondria. Oxygen is required for aerobic respiration, and carbon dioxide is a byproduct of aerobic respiration. So the mitochondria is why your cells and therefore the human body need oxygen at all times, and why cells produce carbon dioxide and the body must rid itself of carbon dioxide at all times. Tissues that are the most metabolically active tissues in the body are teeming with mitochondria. You'll find large concentrations, or large numbers rather, of mitochondria in liver cells, in kidneys, and in skeletal muscles, particularly people who perform a lot of aerobic exercise. Mitochondria are a pretty unique organelle. They have two membranes. They have an outer membrane and they have an inner membrane. And that inner membrane is thrown into these folds called cristae, and those cristae are studded with enzymes, and it is these enzymes that catalyze all of the reactions of aerobic respiration. The cristae, or the folding of the inner membranes, increase surface area to increase the interactions of the enzymes with their substrate. The matrix refers to the space within the inner membrane. It contains DNA. Mitochondria have their own DNA. It also contains ribosomes and all the enzymes involved in aerobic respiration. A very important organelle is the cytoskeleton. And the cytoskeleton is basically a network of structures composed of proteins that extend throughout the cytoplasm. Some of the functions of the cytoskeleton include providing an internal framework for the cell, which determines the distribution of organelles as well as supports the nucleus. The cytoskeleton determines the shape of the cell, it supports the organelles, and it also allows for the transport of vesicles within the cell. And for those cells that move, it also enables cellular movement. So you can think of the cytoskeleton as both the bones and the muscles of the cell. The cytoskeleton is composed of three functionally and structurally different components: microfilaments, intermediate filaments, and microtubules. Microfilaments are the smallest components of the cytoskeleton. They consist of two strands of actin molecules coiled together. They function to support the cell membrane. They are attached to membrane proteins on the cytoplasm side of the cell membrane. They enable muscle contraction in muscle cells. And for those cells that are mobile, they enable movement. The intermediate filaments are composed of long fibers sort of woven together like a rope. They determine the position of organelles, they support the nuclear envelope, and they also form the desmosomes, those anchoring junctions which hold one cell and an adjacent cell together. Lastly, microtubules are polymers of tubulin. These are the largest of the structures that make up the cytoskeleton. Microtubules form mitotic spindle during mitosis. They also allow vesicles and organelles to move within the cell, and they also are responsible for the movement of cilia and flagella. Centrioles are the last organelle we're going to discuss. These are structures composed of microtubules. They're responsible for the formation of mitotic spindle during cell division. They also are involved in the formation of the projections of the plasma membrane, including cilia and flagella. Now let's review the objectives for this screencast. Describe the structure and functions of each organelle. Describe the structure and functions of the three components of the cytoskeleton. The next screencast discusses cell division and differentiation.